Hello. So this week we're going to talk about DNA and RNA and some of the similarities and differences between them and what RNA is used for. But before we do that, I want to go over really quickly DNA replication because I talked about it in my last notes. But I wanted to re-go over it because one of your assignments this week is going to deal directly with DNA replication, and it's important that you understand what's going on. Okay? So once again, cells replicate DNA because they need to divide. And each of those cells that gets produced has to have an exact copy of the DNA that the original cell had. So it does this through DNA replication. So what happens is our DNA is going to get pulled apart. So these two strands, this strand here and this strand here, are getting pulled apart by an enzyme called DNA helicase. And once that DNA strand is pulled apart, another enzyme called DNA polymerase, which in this diagram is yellow and in two spots here and here, that DNA polymerase is going to attach complementary nucleotides to each of the parent strands. Okay, so let's say that in this original parent strand, we had a sequence of DNA that went A, T, G, G, C, A. When polymerase is putting on those complementary nucleotides, it's not going to put on another A. It's going to put on a complement. And you know which complement that is because you know the bonding rules. A is always going to bond with T, and C is always going to bond with G. So if I wanted to put a complementary base onto A here, I'd put a T. If I wanted to put T's complement, I'd put an A. If I want to put G's complement, I'd put a C, C, G, T. Okay? So you see how this strand down here is the complement to the parent strand. It's not the exact same thing, but it's its complement. And the same would be true for down here, because maybe the original parent strand down here was TACCGT, but the complement strand to that one would be ATGGCA. Okay, so that's the strand that would be being made down here, and then this one would correspond to the one up top. It's important that you can produce a complementary strand of DNA using these bonding rules right here, because I will ask you to do that in one of the assignments. If you are still unclear about how that works, please ask me. I'm happy to help you. However, if you just sit there and you don't tell me that you're confused, I have no way of knowing what you're confused about unless you tell me. Okay? So once all that replication is done, we're going to have an end result of two DNA molecules which are identical to one another, and they're identical to the parent strand, okay? Because that's the whole point. We need to end up with DNA molecules that are the same. So let's move on and let's talk about DNA and RNA. This is a little bit of review. How do cells use DNA? Genes are going to be instructions that tell cells how to build proteins. So once again, genes are just instructions for everything that your body does ever. But your body needs to be able to use those instructions, okay? It needs to be able to decode them and use them for things. So the first step to decoding the DNA is to copy a part of the DNA that the cell needs at any one moment, because it's not just going to try and decode the entire DNA strand at once. That would be inefficient. It's just going to try and decode the part of that strand that it needs to do something specific. And that decoding is called transcription. After it decodes this, the strand of DNA, then RNA is going to travel outside the nucleus and go make the protein somewhere, and that's going to be called translation. Okay, so here's a graphic that's actually not starting out at the proper moment. Just give it a second. It'll cycle back around. So here's what's going on. The first thing that happens is that DNA gets yanked apart, an mRNA strand gets built on top of it. Then our mRNA is going to move out of the nucleus of the cell. It's going to go into the cytoplasm, and it's going to travel to the ribosome, where tRNAs are going to bring amino acids, and they're going to start making bonds between appropriate amino acids. Okay? Then, once it's done bringing the right amino acids and bonding them together, then that amino acid chain is going to start folding up, and once it's done folding, you get a protein. So before we get into all the specifics of all that, let's talk about some of the differences between DNA and RNA. As I mentioned before, the sugar in DNA is called deoxyribose. The sugar in RNA is called ribose. And this makes sense because DNA is called deoxyribonucleic acid, whereas RNA is just known as ribonucleic acid. So, whoops, that sugar is quite literally giving each thing its name. Another difference between DNA and RNA is that DNA is two-stranded. So DNA is going to be that double helix shape that you all know and love. That's going to look like this. Whereas RNA, you're just going to see one strand with bases coming off of it like that. Those bases are different as well. In DNA, you should already know because it's on your reference sheet, we have A's, T's, G's, and C's, adenine, thymine, guanine, and cytosine. Whereas in RNA, they're going to be A, U, G, and C, adenine, uracil, guanine, and cytosine. DNA is only going to be found in the nucleus of cells, whereas RNA is going to be found in the nucleus and the cytoplasm because RNA gets the privilege of traveling outside the nucleus. Okay? Why do we need RNA? Why can't DNA just go outside the nucleus? Because RNA is a disposable copy of a piece of DNA. It is a copy of one gene, not all of them. So if you recall, DNA is the blueprints for everything your body does ever. So when your cell needs to do a specific thing, it's not going to want to go and decode every single gene in there because that would, one, take forever, and two, just not be efficient. Like imagine if you were trying to build a bookcase and you had every instruction manual that ever existed. That would be a pain. You wouldn't be able to build the bookcase because you'd spend all your time sorting through all the manuals. Whereas if you just have the manual for the thing you need, you can build something really quickly. And that's exactly why RNA is useful. There's three kinds of RNA that we're going to discuss. The first is messenger RNA. That's the one that's going to carry instructions for assembling the protein. So that messenger RNA is going to carry instructions from the nucleus to the outside. Ribosomal RNA, on the other hand, is going to be part of the ribosome itself. So you can kind of see that over here. There's ribosomal RNA on the ribosome, and that's going to be the site at which that messenger RNA that's traveling from nucleus to the ribosome gets read. Okay? And then last but not least, we have transfer RNA, which is another type of RNA, and its job is to bring those amino acids to the ribosome to start building that protein. Okay, so here's a nice diagram of that entire process together. The entire process is known as protein synthesis, and we're going to talk about a lot about that. But for now, I just kind of want to go over it generically. Transcription is the first part of protein synthesis. That's the part where you have the DNA strand that gets yanked apart just a little bit so that we can make that quick blueprint, that quick RNA transcript of mRNA that's going to then travel outside the nucleus. So the production of that mRNA messenger pigeon is transcription. So to analyze this diagram down at the bottom, we see that the DNA is getting pulled apart a little bit, just in this spot. And we see that RNA, this little purple strand right here, is being built onto one of the strands of DNA, okay? And then after that's done, then that RNA strand is going to go ahead and travel outside of the nucleus. So things you need to know about transcription. 1, it happens in the nucleus. Always, always, always. Transcription will never take place outside of the nucleus. One strand of DNA is going to be used as a template, not both of them, just one, because RNA is single-stranded. Now, that strand of RNA is going to be made following the base pairing rules. Our base pairing rules are only very slightly different for RNA than they are for DNA. Instead of A bonding with T in RNA, A is going to bond with U, and then C is always going to bond with G, just like always. And then the last thing on this slide to know about RNA is that hundreds or thousands of mRNA copies can be made from a single gene. Not at the same time, but if your cell needs to make the same protein more than once, it's going to go back to that same gene and make another copy of mRNA. Okay, so we're going to stop here for now. We're going to stop at transcription, just knowing that the mRNA is made and then it gets to travel outside the nucleus. We'll go over this a little bit in class and then we'll do some stuff with it.